Hello, my name is Baraliakos. I'm a rheumatologist from Germany. In this video, we presented key efficacy and safety data for Bimekizumab in patients with active non-radiographic and radiographic axial spondyloarthritis, or axSpA, from the randomized parallel phase 3b mobile studies. Author disclosure information and acknowledgements are shown here. Bimekizumab is a monoclonal IgG1 antibody that selectively inhibits IL-17F in addition to IL-17A, which have both been implicated in the pathogenesis of axSpA. Bimekizumab has demonstrated clinically meaningful efficacy to week 16 in patients with axSpA in the parallel phase 3 studies B-Mobile 1 and B-Mobile 2. In this video and corresponding manuscript, we present the results to week 52. The study designs of B-Mobile 1 and 2 are shown here. In both studies, patients were randomized to 160 milligrams subcutaneous Bimekizumab or placebo every 4 weeks until week 16, after which all patients switched to Bimekizumab treatment. For brevity, this narration will focus on trends observed in patients who were initially randomized to Bimekizumab. Trends observed in patients who switched from placebo to Bimekizumab at week 16 are described in detail in the corresponding manuscript. In brief, patients in B-Mobile 1 had non-radiographic NR axSpA according to clinical diagnosis and ASAS classification criteria, without radiographic sacroiliitis, while patients in B-Mobile 2 had radiographic R axSpA, fulfilling both body fat New York and ASAS classification criteria. Patient demographics and baseline disease characteristics were similar across treatment groups and were reflective of the active axSpA population. Rapid responses to Bimekizumab were observed for the primary endpoint of ASAS-40 at week 16. Achievement of ASAS-40 response was sustained from week 16 to week 52 in patients receiving continuous Bimekizumab. Around 60% of Bimekizumab randomized patients achieved ASAS-40 at week 52 across the full disease spectrum of axSpA. Bimekizumab treatment led to reductions from baseline in BASTA score between week 16 and week 52 across the axSpA disease spectrum. By week 52, change from baseline in BASTA was comparable between treatment arms. A similar pattern was observed for change from baseline in ASTAS from week 16 to week 52. Almost all patients had high or very high disease activity. This means an ASDAS score of equal or higher than 2.1 at baseline. By week 52, Bimekizumab treatment resulted in a majority of patients achieving ASDAS low disease activity and around a quarter of patients achieving ASDAS inactive disease. Rapid reductions in CRP observed at week 16 were sustained to week 52 in continuous Bimekizumab treated patients. Reductions from baseline in SPARC sacroiliac joint scores were observed between week 16 and week 52. The mean change from baseline was comparable between treatment arms by week 52. A similar pattern was observed for the Berlin Spine score. Focusing now on safety, by week 52, at least one treatment-emergent adverse event was reported in 75% of patients with NR axSpA and 75.5% of patients with R axSpA. No deaths occurred in either study. As expected, with the known safety profile of Bimekizumab, an increased incidence of fungal infections was observed in Bimekizumab-treated patients. However, all were non-systemic and localized. To week 52, IBD incidence was in line with the background rates for axSpA, while uveitis was reported in 3 patients with NR axSpA and 7 patients with R axSpA. To conclude, dual inhibition of IL-17A and F with Bimekizumab led to sustained efficacy across the full disease spectrum of axSpA over the 52-week period. Consistent responses were observed between Bimekizumab randomized patients and patients switching from placebo to Bimekizumab at week 16. The safety profile was consistent with the known safety of Bimekizumab.